All right, hi online class. I just asked the class the clicker question that's here on the board that when you burn fat and lose weight, where does that matter go? We're going to talk about the options. You'll see here on your slides, if you want to do a similar discussion, this is a TED Talk that has a scientist asking people the same questions. It's that YouTube link. Okay, but answers here, I think I'm sneaky, right, with sneaky answers. I was actually surprised. I thought a lot of you would say it leaves as you poop, right? Your body does get rid of waste, right? But you all are right that the waste that's leaving as feces is waste that you haven't pulled into your body yet, right? I mean, and yeah, like some nitrogenous waste is going to be leaving as in urine, right? But in general, the stuff that's like part of you, that's not the main way to get rid of it. Okay? Cool. So yeah, the idea is, you know, you weigh 200 pounds, you lose 10 pounds, that weight had to go someplace, right? Go someplace. I get that people like the idea that it's changed into heat, because yeah, as you exercise, as you move your muscles, you're going to generate some heat, right? But does heat have a weight? Does heat weigh anything? No. So if you have a blob of matter, right, some amount of matter, just because things got hot, that doesn't account for the weight, right? Like the individual carbon atoms, heat doesn't take away individual carbon atoms. Carbon atoms weigh a certain amount, and we're going to go with carbon atoms. Most of what we're talking about is carbon atoms, right? Carbon and hydrogen. Mostly carbons, right? So no matter how hot you are, it can't be that. Right? Just getting hot doesn't make you lose weight. Right? Similar idea with muscle. If you have some sort of carbon chain, right? Carbons, 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 carbons. Even if you were to rearrange those into some other form, right? There's originally carbons that were in, say, lipids. Now you, series of steps, end up with that carbon in protein. The carbon still weighs the same amount, right? So that can't work either. Right? This, just for the record, this question of where does weight go when you lose weight, you will catch most people out. I mean, I don't know, but ask your roommate. They are going to get it wrong. They just will. But holding that thought for a second, air has a bunch of gases in it. But in general, when you breathe in, what are you trying to get in? O2, yeah, oxygen, right? Deep breath. When you breathe out, what are you breathing out? CO2, carbon dioxide, right? Okay, so carbon dioxide is CO2, it's oxygen, right? So you breathe in O2, you're breathing CO2 out, so O2 you're breathing out still. Where did the carbon come from that you're breathing out? If you breathe in oxygen, it was where? It's already where? You have some inside of you. You're not. Yeah, yeah. So, I don't know if the back could hear that. Okay. So yeah, you're breathing oxygen in, right? As you breathe the carbon dioxide out, the carbon's coming from inside of you, right? So oxygen in, carbon dioxide out. The carbon had been there. It's kind of like if you are you're watching a house, right? And you see one person go in, and then two people come out. You're like, oh, I'm guessing that other person was in the house originally. Yeah? So for the answer for this one, when you burn fat, where does that, and you lose weight, where does that matter go? Most of it's leaving as you exhale. Just so you know. I know, right? This is not to recommend that, like, my weight loss technique is, don't do that. You're just going to make yourself dizzy, okay? But yeah, you're getting rid of the carbon that you break down. A lot of it, you're getting rid, most of it, almost all of it, you're getting rid of as carbon dioxide. And today we're going to see why and how. But before we go into that excitement, I need to tell you about an assignment that's coming up. Come on. Come on. Hi, little computer. You can do it, computer. Alright, so, right before you leave for Thanksgiving, you have an essay due. It's like the big essay project in this class. Do it, it's worth a lot of points. Don't panic about it, right? Like, this doesn't need to be your life's work. Just do it. You're going to have a rough draft due end of next month, okay? So, it's fairly open-ended with some constraints. So the deal is you're going to find some biological topic you're interested in, right? Something that you're like, I care about this. Humans, animals, plants, fungi, mold, bacteria, the coronavirus, whatever, right? Something that you're interested in. You're going to get good scientific information about this. Your cousin is bad scientific information about this. They might be brilliant, that's great, but you need something peer-reviewed, okay? You get 3 sources. If you have any questions about this, I'm happy to check your sources, okay? If you're like, huh, I saw it on the back of a cereal box. It looked official. They had, like, fun facts to know, and I used that. Feel free to ask me, I'll be like, nope. And there are some things that look really scientific, and you can still use them as sources, but they can't be your peer-reviewed sources. Say, like, your textbook. You should believe most of what's in your textbook. It's not a peer-reviewed source. It's good. You can definitely cite it, but that doesn't count as one of your 3. Then you're going to write an essay that looks sort of normal, somewhere between 5 and 10 pages, double space, 12-point font, normal margins. If you want it longer than 10 pages, people have done this before, go for it, but that's probably about the length we're after, okay? If it's less than 5 pages, 5 pages double spaced isn't that much. You probably won't be able to address the question, okay? Things this is going to include. I care that you write in English, and I care that you write something that's readable, but format's kind of up to you. Some sort of introduction, background, evidence, conclusion is a really good way to organize this. But the things that your paper absolutely has to have. You can describe what the topic of debate is, right? Some sort of background on the context or the controversy. What research has been done? What's known? Like, what's going on here? You must include some sort of qualitative information, numbers or graphs or charts, something that's like, yeah, this, instead of just being like, it's really important to wash your hands because it decreases the likelihood of disease. You can then throw a chart in that's like, look, incidence of disease in people who never bathe or whatever, right? And you're going to have some version of what's the science backup. Yeah? Couple caveats here, which just to make things better, more interesting. It's a biology paper, not an opinion paper. Okay? I'm glad you all have opinions, and this is good. You should have opinions. And there are things that are super, super important. Questions like, should abortion be legal? Is a really important topic, right? Or, is socialized medicine right for us? Or having universal access to healthcare? Really important topics. They deal with biological topics, but they're opinion-based, okay? So, some question like, do annual doctor's visits increase life expectancy? Totally cool for this. Completely fine question to address, right? Saying, do we need to have a single-payer healthcare system? That has too many other things going on into it. Does that make sense? I'm not saying it's not an important topic, but you need to keep it focused on something you can address scientifically, okay? So some things that have happened in the past that are pretty good examples of this. Is fluoridating water good for health, right? You could read the evidence for that, present the, like, yeah, you know, some people say no, some people say yes. The science says thing, right? Are we creating super germs? Is running without shoes helpful, right? Like, does it lead to better outcomes? Does microwaving food change it nutritionally, right? This kind of idea. Concerns about this? You can do it on basically any topic that's biological, and pretty much any topic you could have be biological. do have something that there's enough research about that you can like look something up, and do have something. I mean, this isn't a requirement, but if you care about it at least slightly, it makes this way more interesting because you're like, hey, I learned something. Yeah.
We have to write it as a question that needs to be answered.
No. Like, what do you mean?
Like, if you were writing about tangent photosynthesis, could you just write about it instead of having a question about it?
Yeah, sure, but there's going to be, yes, you're absolutely correct, but there's probably going to be, like, you're not writing a textbook, okay? Don't be like, the main process of photosynthesis are photosynthesis 2, then photosynthesis 1. It bugs me that it's 2, then 1, anyways. It's in that order. Yeah, but you'd be able to, because, like, people have long wondered the importance of electron transport chains. Yeah. Totally good. Yep. Other questions? Yeah. It'll be good. I actually kind of like reading these. Okay, so, like, I feel sorry for myself because I'm going to grade them over Thanksgiving. And I'll, like, pout. Not about you, but, like, I have to grade them. But they're actually usually pretty interesting. Because people come up with some, like, I hadn't thought about that. And they look it up and they figure it out and it's good. Yeah? Okay. Please, 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 please. Have it be scientific, okay? I speak from reading some of these and then it's like, If you cite a book of scripture, I'm totally down with people reading scripture, that's good, but please don't cite a book of scripture in this, okay? It's a different thing than what we're talking about. Yeah? I feel weird about how to grade this. Okay. We're good? On to today's topic. This was, but just pushed it at the beginning because it's hard for online folks to have the break in the middle. Okay. We're going to come back to that, the carbon leaving as you exhale. Sounding probably like, I don't know, your grandpa maybe? Life is work, right? Okay. It is, though. For life to exist, you've got to be able to have meaningful work happen, right? Be able to cause change, okay? This means you're going to need energy from outside sources. And we've got this whole circle of life situation going on here, which is described in this chart here. You've got energy coming in as light, right? That energy is going to be able to, with the help of photosynthesis, be able to have carbon dioxide and water be changed into organic molecules. You just rearrange them into organic molecules plus oxygen. These organic molecules are now holding a lot of energy in them, right? So energy flowed into this, built a complex organic molecule, a lot of energy there. You're able to use those complex organic molecules, break them down. As you break them down, you can harness some of that energy to make ATP, and then you're able to use that ATP to do other work. Yeah? As it's happened, you've broken down this organic molecule. You're going to, with organic molecules and oxygen, you're going to end up with carbon dioxide and water. But then with light energy, again, you can put it back. So far, so good? I really should play the Circle of Life like Lion King song. I need to do this more often. Here's this video. The production quality is questionable, but it's a nice overview.
Could a carbon atom that was once part of Albert Einstein be in you? All living things need to take in carbon-containing molecules to fuel their activity and build their cells. This is true for busy students and for all organisms. Let's look at how carbon atoms move through the carbon cycle in the Arctic tundra. Carbon atoms from carbon dioxide are incorporated into living things by producers, such as this plant. Carbon dioxide enters a leaf, moves into a cell, and goes into a chloroplast. There, in the process of photosynthesis, carbon dioxide is used to make sugar molecules. The sugars can be used to build larger carbon-containing molecules, carbohydrates, lipids, proteins, and nucleic acids. In this way, carbon that was in the air is now part of the plant. When a plant is eaten by an herbivore, or primary consumer, carbon in plant tissue enters the primary consumer's digestive system. There, plant molecules are broken down. These small molecules are then used by the primary consumer to make larger molecules for building its own tissues. In this way, some of the carbon that was in a plant becomes carbon in a primary consumer. Similarly, when a primary consumer is eaten by a higher level consumer, some of the carbon in the primary consumer is incorporated into the higher level consumer. Carbon-containing molecules in dead plant material, animal feces, and dead organisms are taken up by decomposers, such as fungi and bacteria. Decomposers break down these materials and incorporate some of the carbon atoms into their own bodies. We have now seen how carbon moves from carbon dioxide to producers, to primary consumers, to higher level consumers, and to decomposers. How does carbon get back to the atmosphere? During cellular respiration, carbon-containing molecules are broken down, and ATP is made. Carbon dioxide is released as a byproduct and returned to the atmosphere by producers, consumers, and decomposers. Overall, this return of carbon dioxide by cellular respiration is closely balanced by its removal from the atmosphere through photosynthesis. The global carbon cycle involves living things on land and in rivers, lakes, and oceans. And by the way, considering the huge number of carbon atoms in each person and how quickly carbon atoms move through the carbon cycle, there is almost certainly a little of Einstein in each of us.
Yay. So far, so good? Okay. So, to get to that point of where we're getting carbon off and doing stuff, there are a couple things that need to happen first. So, they're going to be, remember metabolism from last week? Yeah, okay, great. We're now looking at some catabolic pathways. So, catabolic pathway, building up or breaking down? Breaking down. I heard somebody say it, but I don't know who, but yes, you're right. Breaking down, right? So catabolic pathways, they're going to break down organic fuel. So that could be something like protein. It could be something like sugar. And they're going to release energy that's stored there. Basically, in these organic fuels, there's some really high energy electrons, right? Electrons that are enthusiastic, happy to be there, have really, really high free energy. The cell's going to take those off and move them around, and we're going to be able to harness some of the energy that way. So, couple terms, we're going to get to them more later. The breakdown of organic molecules gives off energy, okay? Complex, beautiful molecule, you tear it apart, it goes from high free energy to lower free energy, okay? You can have fermentation occur. Does anybody know a process that, something that uses fermentation? Yeah, yeast. So, if you're using yeast to ferment something, it's going to be fermenting. There's another hand. Somebody else said something? Alcohol, right? Alcohols you're going to ferment as well. Yeah. So, if you're making bread, right? You're running this, or you're rotting anything. You're going to have the sugar going to get broken down, but it doesn't need to use oxygen to make this happen. It'll just go for it. All right? Aerobic respiration is going to consume organic molecules. Whereas fermentation is just going to sort of break them down, aerobic respiration is going to completely break it down. Yeah? And it's going to take oxygen. So if you think of aerobic exercise, takes oxygen, right? There's a link there in case you want to go to it, to Richard Simmons' Sweating to the Oldies full workout video, if you're interested. It's aerobic respiration. Takes takes oxygen, right? Yeah? I was going to show a clip of this in class, and then I thought better of it this morning. I'm like, maybe not. But it's there if you want it. When we're talking about aerobic respiration in a cell, we're not talking about, like, the cell has little tiny lungs and is breathing or something, okay? But what we're talking about is the cell taking oxygen in to drive some of these breakdown processes. Yeah? Okay. Here's the pathway for this whole week. If everyone's okay with this, we're going to pretend that it's just sugar. So the cell can break down lots of different types of molecules. It can break down proteins, it can break down fats, it can break down a bunch of things. It can break down complex carbohydrates. We're going to go through glucose, because glucose is the most straightforward one of this. It's a simple sugar. It's kind of the default, right? So if you have something like sucrose that you're breaking down, you're going to first tear it into fructose and glucose, then break it down. Anyways, we're going to watch glucose getting broken down. Similar pathways for the other, but you have to do a few more things. So, okay, here we go. We're starting, actually, because I'm being recorded, I'll be right here. Okay, this thing over here is glucose, this C6H12O6, right? Glucose plus 6 molecular oxygen, so 6 O2s, right, is going to go to carbon dioxide, water, and we're going to get off energy from this. So on this side of the equation, there's energy here in the high energy electrons in the sugar. On the other side, we've liberated them. We're going to use that energy to make ATP. We're going to use that energy, it's going to make some heat as well. Yeah? Walk me through this with making sure I didn't lose any chunks of anything. On this side, when you start everything before this arrow, how many carbons do we have over here? 6. On this side, yeah, so we've got 6 carbons right here in the sugar. How many carbons do we have on this side? 6, right? Because we've got 6 of these CO2 molecules, right? Let's look for hydrogens. We've got 12 hydrogens here. How many hydrogens on this side? 12, right? Because we've got 6 of these H2 and H2O. Oxygen is the tricky one to watch. We have 6 plus how many? 12, right? And look, we have 12 plus 6 on this side. Do we see that? We didn't, everything's accounted for on both sides, right? We went from complex to less complex, and we released energy in the process. Yeah? Okay. The steps for how this happens are beautifully organized. They really are. Traditionally, or last year, students either really like this section because it's like lots of little pieces moving and interesting ways they fit together. And then some people really hated it because it's lots of little pieces moving, somehow they fit together. Yeah? Before we get into how this is actually happening, we need to talk about Mondays. Sometimes, okay, often it's hard to get going on Monday for me. Because, like, I like teaching, I like being here, it's great, but, like, the weekend's nice. And so getting up, I'm like, ugh, it just takes me a minute. And I live in Columbia, which is a ways from here. It's like half an hour, it's not bad. But I drive a car, and I was thinking about how, if I wasn't worried about legal requirements, normally when I drive my car, it burns a little bit of gas at a time, right? Like a little bit, a little bit, a little bit, a little bit. Very controlled reaction. Just thinking, if I just opened the gas tank and like threw a match in there and had it all explode at once, I could go so much faster, right? I'd be like, boom, instead of slowly. No? You're saying no? Anybody else think it's a bad idea? I'm not worried about the legal requirements, but just the like, boom, I get here fast. Okay, people are thinking it's a bad idea? Thanks, yeah, okay. The front row apparently wants me to live, back row wants me to live. Thank you, I appreciate this. Okay, yeah, you can't actually harness it. If you just explode the gas tank, don't do this. If you explode the gas tank, you'll get some energy off, right? But you won't be able to harness it, right? Because just boof. Okay, is everyone okay with that? using the same amount of fuel, it's like a 12-gallon tank, using the same amount of fuel, if I burn it a little bit at a time, I can get a lot more work done. I can get a lot more change, a lot more movement than I could if I just try to have it all happen at once. Fair with that? Is everyone okay with that idea? The cell is going to do the same thing with its breakdown of complex organisms. So it's got a complex molecule. It's going to use a sugar, and it's going to, in a series of controlled steps, pull the energy off. If it all happened at once, there's no way to harness that energy. Like, it's just too much. Honestly, if all the energy in like a Snickers bar was released all at once, there's more energy in a Snickers bar than there is in a bolt of lightning. And I'm not being dramatic. Like, there really is. Like, you would just die. Okay? It comes off in a very controlled series of steps. Alright? That's not my car, by the way. But I liked it. Okay, so this is the idea. You start with some high energy electrons that you're pulling off of things. You're going to go to low energy electrons. But if you do it in a series of steps, you can harness energy off each series of steps as opposed to losing it. You'll still start at the same place, end at the same place, but you get more usable work. Nods? Okay. I do appreciate that class is like, yes, please live, as opposed to try it and see what happens. I actually do know better. Alright, so conceptually, everyone's with me? Doing okay? We're going to use a series of controlled reactions. The ones we're going to care about right now are going to require oxygen. We're going to series of controlled reactions using oxygen. We're trying to get some of the energy out that was put into sugar. Alright? 3 main steps. So to get energy from glucose, 3 things, lots of things, but 3 main steps here. Okay? We're going to have glycolysis. That's going to happen first. Remind me what that list means, like with hydrolysis, a lysosome. What does that mean? Yeah, breaking down, cutting, right? Okay. We've got this glyco part. I need to not wander around. Glycolysis. It's going to break down sugar. It's going to break down glucose into 2 molecules. First step, just snap glucose into usable chunks. Yeah? Snap. Next, we're going to do the citric acid cycle. Pyruvate oxidation and citric acid cycle. It's going to finish breaking down what used to be glucose. Start with a glucose molecule, 6 carbon longs, breaks it down to 2 easily held chunks. You're going to finish breaking that down. As you tear that to bits, you're going to pull electrons off of it. The carbon's going to leave as carbon dioxide, but you've harvested a bunch of electrons. You've gone through all this work to bust up this organic molecule. At the end of it all, you're going to do oxidative phosphorylation, which is basically the payoff stage. That's where you're going to make a bunch of ATP. 3 main parts. Your book has them color-coded throughout the whole book. It has glycolysis in blue. The middle step, that citric acid cycle, pyruvate oxidation, comes up as orange. And then purple is oxidative phosphorylation, just to keep things clear. Also, if you've learned this before, or old textbooks, often called the citric acid cycle the Krebs cycle. Same thing, just called a different name. Great. Here's the overview of what's happening. We're going to look at things in depth. Like, I'll talk about nothing else this week. This is the mitochondria here, right? Glycolysis, this first step, we've got glucose coming in, pyruvate coming out. So we start with glucose, we're going to change it to pyruvate, right? Bust it up. Doing that, we're going to be able to get some electrons. Not to worry about that. We're going to get a little bit of ATP. Not much, but you do get a little bit. Then you're going to shove pyruvate into the mitochondria. You're going to change it slightly. You're going to go through the citric acid cycle. Again, you're going to get some electrons off. That's great. Again, you'll get a little bit of ATP. Not super exciting. The exciting bit, the exciting to some people bit, is this last part, the oxidophosphorylation, where you finally get to use these electrons you've been pulling off the whole time. These high energy electrons, you've been pulling them off, and now you get to use those to drive ATP production. Yeah? Great. Deep breath. Everyone's doing okay? Okay. I have a video here. It's going to talk us through this whole thing. All right? I realize there are lots of moving parts. So right now, you're just getting kind of familiar with the moving parts. We will then spend more time than you want to talking about each of them. Please play.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy and fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, 1 carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, The flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
Sweet. Lots of parts there. We're watching it. A couple things to notice. Did you all see how carbon's changing form several times? Well, not carbon. Carbon skeletons are changing form. You start with the sugar. It keeps things happen to it. Things get added, things get removed. But overall, it's getting smaller and smaller. You're breaking off carbon bits, right? You also saw how electrons are being pulled off, right? And yeah, there's some very a controlled series of reactions taking place. So far, so good. Also, are people comfortable with the difference between mitochondria and mitochondrion? It's like you have mitochondria, lots of them. You have dice, or you have one mitochondrion, a die, right? It's just the plural. Okay. We're good? Oxidative phosphorylation, powered by redox reactions. If you're in chemistry, great, this makes sense. If not, we're just going to move on. This is where most of the ATP is going to be made. We'll get to that later. About 90% of the ATP is made there in the mitochondria. You can make a little bit of ATP during glycolysis and citric acid cycle. It's not very much, but overall the whole thing from glycolysis through oxidative phosphorylation, for one glucose molecule, depending on the conditions, but almost always you'll get about 32 ATPs off. Okay, you'll get a bunch. And your little mitochondria just keep running around the clock. So that's good. Okay, I realize we have like 6 minutes left. 10 minutes left, we have 10 minutes left. Oh, good. Well, okay, that clock, it's a little bit weird. We're going to go through a 10-minute concept, which is we're going to start looking at this glycolysis part, okay? Glycolysis is part of the whole process, but now we're just looking at this glycolysis part, okay? Yeah? So glycolysis is this first step in this process. It's the sugar splitting part. It's happening in the cytoplasm of the cell. So you're inside the cell, but you haven't got to mitochondria yet. It's just inside the cell. And the main goal is to break down glucose into 2 molecules of pyruvate. All right? Glycolysis does not notice or care if there's oxygen. Doesn't make a difference. With oxygen, without oxygen, it can just do its thing. There are 2 main portions of this. There's an energy investment portion and then a payoff portion, right? So it looks like this. This is the overall picture of it. We're going to go through each step. This is the part to know, okay? We'll see this at the very end. Here's what's going on. You start with glucose. Here's our beautiful glucose molecule. Yay. Using ATP, you phosphorylate this thing. There's a whole series of steps here. If you're in, probably in biochemistry, you'll memorize this whole beautiful thing, and that'll be fine. But for our intents and purposes, you're using some energy to kind of to change this glucose molecule around a little bit. You then have fructose 6-phosphate. It's a very nice little molecule here, because you've added energy. There's been an enzyme that's helped change this. From that, you're going to add some more energy into it. You're going to use another molecule of ATP. This is now your second molecule of ATP you've used here. And you end up with this double phosphorylated molecule. That, then you have additional series of reactions. You're going to be moving stuff around. And breaking things up. You go from having this ring structure to you busted it up open like that. You then, from there, are going to pull off some electrons. You're going to end up with three carbon chains, have a few things on them. And then a series of more steps, you're going to pull off hydrogens and oxygens. No, there's hydrogen here, there's no hydrogen here. See, hydrogen here, no hydrogen here. There's an OH here, no an OH here. Right, you're pulling that off as water. And you, at the end here, you're going to, you have two ADPs plus some inorganic phosphates. So you've got an inorganic phosphate film molecule, you have ADP, you're pulling them off, you now made ATP. And this is the overview. Okay, so those steps are just so I don't lie to you, so you kind of see what's actually happening. The take-home message is this. You start with glucose, right? And you're going to use ATP, which seems a little weird, right? Don't we want ATP at the end? You have to pay a little bit in to get started, okay? You pay 2 in, but in good news, it's not a problem because you end up making a total of 4. So you end up having a net 2 ATP produced, right? You paid 2, you got 4, so you overall made 2 new ones. For our carbon chunks here, we started with glucose, and now we ended up with 2 pyruvates. All right? So glucose, went from glucose to 2 pyruvates plus water, right? Overall, we ended up with 2 ATP, and we went from having some empty electron carriers and having some, sorry, I'm too short for this, let's see. From having electrons in the glucose, we ended up pulling some of those off, and we now have electrons hanging around in electron carriers. Questions about that? There's kind of a lot going on there, I realize. But the important bits, glucose, costs some energy, get more off. This molecule still has a lot of energy in it, this pyruvate. There's some energy here in the electrons that are living in here. And you did make some ATP. So overall, it's a worthwhile thing for the cell to do. Everyone solve where we're at? All right, that's all we have for today. Unless there are questions? Great. In that case, I will see you on Wednesday.